Hello students, welcome to EPG Parsala. I am Dr. Deepak Kumar, Assistant Professor in the Department of Biotechnology Molecular Medicine, PGIMS, Rohtak. Today we are going to discuss about the module Gene Knockout and Targeting under the paper Animal Cell Biotechnology. So the gene knockout is that we do not want the gene. The one thing is that if you want to know the importance of anything, like how much this is important, why this is important, what is the function of this, the best thing is that to remove that from the place. Like in the body, when there are so many genes, if you want to know the gene, function of the gene, the simple thing is that we will just remove that gene. Like in your home, if you want to know the importance of a person, if the person is out from your home, you will know that how important the person is. The same thing, the gene knockout, if we knock out that gene, we will know that what is the important, what is the physiological changes, what are the phenotypical changes, what are other changes, like whether it's involved in the disease, whether it's involved in the gene interaction with the protein, with the with the drug. So we can we can understand all these things, that where the gene is important, because we know the sequence of a gene, we have sequenced it, we also translate the gene is functional and it's producing the protein. In that case, you don't know what is the function of that protein, what is the function of that gene. The thing is, you should remove it from the body, then you will know that the function of that gene. So for that, we need to prepare the knockout animals. We need to knock out the gene, or we need to target the gene. If we will target the gene, we will also know that the gene expression is reduced, and when gene expression is reduced, what are the changes. So this is a very important thing, and that's the reason we need to go for the gene knockout, and we need to have the understanding of gene knockout, being a student of medical biotechnology or student of biotechnology. So what are the learning objectives of this module is that first we will know what is the gene knockout or targeting, and how to knock out a gene. And third is, we will also discuss what is the conditional knockout animal and why it is so important, and we will also discuss the application. So we need to knock out a gene, we need an animal for that. Okay, so there is chance that we need to select an animal. So there are chance of, like, we can use mouse, rat or rabbit or other mammals, but the thing is that we need a animal which have low cost of maintenance, and the generation time should also be less, and which make the mouse as a favorite animal model, because it has very low cost of maintenance, and generation time is just 9 weeks. And the one thing is that the mouse and human similarity of genome is in the DNA is about 97%. Okay, and another good thing that the molecular stem cell technologies also allow the researcher to create the custom based mouse through the gene targeting in the mouse embryonic stem cells, because within 9 weeks you can have another generation. Okay, so this is I'll not say that is an easy thing, but I mean the concept is easy, but thing is very tedious. So that's why we need an animal which can be used for the purpose for our purpose, and which can be also timely favorable, because if we'll take we will take so much time to get a next generation, then we'll not get the results. The one more good thing is that in the mouse, that the human disease like this cardiovascular disease, cancer, diabetes, we can mimic these diseases in this mouse model, and we can also induce the like cystic fibrosis, Alzheimer in the mouse genomic environment. So when we have such models, such disease models, that will help us a lot to understand that how that how the genome is changed, and how that the cause of the disease, that these are the genes which are responsible, and these are the, and how we can treat this disease. Next. So gene knockout means we have to completely exclude a gene. Completely exclude means that we have to exclude a gene in every part of the body, like brain, liver, or any tissue, or kidney. It should not be like that, that gene is present in one part, one organ, not in the other organ. So that will not be the gene knockout. For gene knockout, one must remove the gene completely. For that, the gene need to be replaced from the other gene, from gene of your interest. And in that way, you can use a vector where you can use a resistant gene, and then resistant gene, like in this you can see the gene here in the blue color, and the vector where the gene is resistant gene, which is coded here, and so that resistant gene can be replaced with that gene. In that way, the gene which is the endogenous gene, which is completely removed, so that's why resistant gene is incorporated in the genome. So gene targeting, it is little different than the gene knockout. Like in gene knockout, we have knocked out a gene, but here we will target the gene. In target the gene means, like, we will target the transcription, and we will reduce the protein expression. For that, we can use the RNA technology. The RNA technology, small fragment, RNA fragment, can bind to the their complementary DNA. They are the 21 nucleotide, 19 to 21 nucleotides RNA fragments, and then they can degrade the transcription, transcript the RNA, and therefore that is also reduce the translation. So then the amount of protein will reduce a lot, like up to 90%, 90 to 95% cases, the protein is reduced. So the thing is that if we have to knock out a gene, the thing is, if we have to do like so many experiments with the same gene, we have to do so many experiments, then we have to knock out the gene. But if we have to just do small studies with a gene, then we have to, it's better to target the gene by gene knockdown or gene targeting. So next thing is the transgenic. Transgenic is little different than a knockout, where we introduction of foreign gene or alter from an introduction of foreign gene, or we can alter the endogenous gene into the organism. Okay, and generally it does not replace, but rather to insert an additional copy of the gene. So the gene we introduce is called the transgene, and the organism as transgenic organism. So how to knock out gene from animal? So as we have to exclude the gene from the whole organism, then we have to target the gene at embryonic stage. If we are able to target the gene at embryonic stage, then only we can knock out the gene from the whole body. What we need to do for preparing a knockout mouse, what we need to do, this is the fertilized egg from male and female, and after fertilization, at the blastocyst stage, when there is a mass of cells, the high density of cell mass we can see in this picture is of a light blue color, and high density of cell mass, you can have to take it out the cells from there, because this is the embryonic stage. Each cell has the capability to generate a full organism, a complete organism. So what we need to do, we have to take out the cell from the high density mass of the cell, and then we have to grow in the culture media, so that this every cell has the capability to grow in the organism. So if we target any gene in these cells, it means the gene is completely knocked out. So what we'll do, we need a vector for that, like like in this case, this is the red color, you can see the neomycin resistance neomycin coding gene, and two side yellow color, these are the recombinant sites, homologous recombinant site. So what we we have to find our gene of interest that need to be replaced. So for the to find a gene of interest, it should be somewhat similar to that one. So for the homologous recombination, it should not be like that any other sequence, because it may cause some very weird phenotypic or physiological effects. So what the thing is that we have to prepare a vector, and that vector need to be incorporated through the electroporation. So during electroporation, we have to impose some electrical current. Electrical fields are induced by high voltage current for some time. In that case, the membrane of the cell will open for a while, and then that vector or DNA can get into the cell, and after that the cell get healed, and your gene of interest is inside the cell. So these genes, this vector when go inside the cells, then there is a process of homologous recombination. What is happening in the homologous recombination, that there is a exchange of strand between the vector and the genome. So in that case, as you can see, in the homologous recombination, there is an exchange of strand, like the D loop side. You can see that the sky blue strand is exchanging with the red one, and that's why the strand invade another strand, and that's how this is as per the Holiday junction. You can see that the strand get exchanged. This is a crossover by that the strand can get exchanged between the vector and the genome. So that's how in the body also, in the complementary side, the homologous recombination took place, and that's why your gene of interest can incorporate in the genome, and the endogenous gene can exchange with the vector. So when the gene is exchanged, in that case the endogenous gene is completely knocked out, is completely removed from the cell, and it is exchanged with the vector. So what do you find after homologous recombination? There is a chance that is a 10% or 20% of cells go through the process of homologous recombination, not all the cells, and there will be very few cells that go through this process of homologous recombination. So what will happen after completing this process? It means the gene has completely removed where the homologous recombination took place. Now the next step is to select it. How to select it? That which of the cells go through the process of homologous recombination. So as we have exchanged the gene with the neomycin resistant gene, when we grow this cell in the neomycin containing media, what will happen? The cells which do not have the neomycin resistant gene will automatically get killed with the antibiotic effect, and the cells which have the neomycin resistant gene will only survive in that media. So after selection, we will have the cells in our plate which have the neomycin resistant gene. It means the gene of interest has been completely knocked out. What we will do, then we will transfer these cells into the mouse. So one thing, first we have took the cells, embryonic cells, that is the donor mouse, and then here where we are incorporating the cells is the recipient mouse. So these are two different mouse. Okay, so when we incorporate these cells as in the recipient mouse embryo, then what will happen? Then we can cross our finger, you can wait for the time, and let's see, you can hope that some of them can develop into the knockout mouse. So we have to wait for this, and it may happen like some of the mouse may have the gene knockout took place. So what happens that some of the mouse which are developed maybe knockout mouse, but it may not be the completely knockout. It may be chimeric mouse, where that it's not the completely replacement like chimeric or mosaic mouse. Then what will happen? Then we can cross these mouse with the wild type mouse, and we can found at a next stage that some of the strains, some of the mouse have some of the chimeric mouse, some of the wild type mouse, and at the next stage, when next step, then we will cross these two chimeric mouse, then we may found a strain that you can see here, like white mouse. This is completely knocked out. I have shown is just for the representation that the wild type mouse is like a black color and is little light color, when the chimeric mouse, and when completely knocked out, is completely lost the color. So conditional knockout, why we need a conditional knockout? The thing is, the constitutive knockdown of mouse sometimes may produce some undesirable result, or maybe embryonic lethal. Therefore, there is need of conditional knockout mice. The thing is that when we knock out a gene at the embryonic stage, sometimes the embryo is failed to develop. It means that the gene has some importance in the embryogenesis. It means that we cannot knock out that gene at the embryonic stage. As I in the talk, I started with that we have to knock out the gene at embryonic stage to get a knockout gene, knockout mouse or knockout animals. The thing is, the conditional knockout is little different, because we cannot knock out the gene at embryonic stage, so we have to knock out gene at later, at as per our experiment or as per our interest or as per our study. So the conditional knockout, what is that? Conditional knockout enable the inactivation of gene of interest at specific tissue or a specific point of time. Like sometime that we have to knock out the gene at this point when we induce the drug, or we have to study the gene-gene interaction. We have to incorporate some protein in gene, some hormones, their effect or their impact on the disease we want to study. So at the gene which are interacting with those metabolites, those drugs, so we have to conditionally knock out them, not at the embryonic stage. So in some conditional knockout, the gene is knocked out at specific time or point. It is not like whole body. The gene is knocked out from the whole body. It's not from the whole body. It is from the specific tissue. In that case, the other tissue of the gene will retain its wild type function. The gene will completely functional. Like if we knock out from the brain, then it is just knock out in the brain, not in the other part of the body. Like that in the gene knockout, where the gene is completely omitted, completely removed from the whole part. When conditional, it is just from the, let's say, in the specific tissue. And the one good thing is that the Cre-loxP system, which is most commonly utilized for conditional mutation of a gene or conditional knockout of the animal. Pronuclear injection to the transgenic mouse, it is like the site integration of the injected DNA into a genome is random, because what we'll do, we will micro inject the foreign DNA into the fertilized egg. It is better that as soon as we will transfer the foreign DNA, there is chance of recombination is more. What will happen? The number of copies we inject to the DNA is random, and in single insertion, about more than 100. The range of foreign gene expression, range of phenotype. Like if there is a transgenic gene which is expressing, then there is chance of change in the phenotype. So it is like we are inserting a gene to prepare a transgenic mouse, and it is a little different than the knockout, because here we are in a knockout, we are removing the gene. Here we are incorporating the, like if we will transfer the foreign gene in a 2-cell stage, then there is chance that about 10 to 30% of the germ cell lines may have there is chance of transgenic animal. But if we will transfer it at the modular stage, like 8 cell stage or 16 cell stage, there is chance that we hardly get like 10% of the chimeric mouse. So what are the applications of generating a knockout mouse or knockout animal? The thing is that the gene knockout helps us to do the difference between a normal and knockout organism, because whenever you knock out gene, there will be some changes at the physiological level or the phenotypic level where you can visualize them. Okay, and you can identify the disease causing gene also, that what are the genes. If you knock out a gene and it causes a disease, then it means that that gene, if there is a defect in the gene, that disease, it can cause a disease. So this is also helpful in the identification of disease. It helps in the investigating the pathway, like cellular pathway, immunological pathway, or inflammatory pathway, etc. It also helps to study the gene function in the disease condition, and it's also help for the cure of the disease. Like if in a disease condition, if the gene is lost and the mouse produces particular disease, then we will completely understand that this gene is responsible for the disease. So as I said that due to the constitutive knockdown of a gene, there may be some problem, like the embryonic, some of the genes may be can cause the embryonic lethal, and or some weird effect that we will not able to the study our desire, we cannot conduct our desired experiment. So there is concept of conditional knockout. Conditional knockout means like we are not completely knock it down, but we are knocking down in somewhere, like it's either tissue specific or some inducible system. So there is a Cre-loxP. This is the most is the most useful system. And what is the property of this system is that when you will put a gene sequence between the two LoxP site and cross with the Cre-recombinase, then the gene is get omitted, the gene get removed. So what is LoxP? LoxP are the like 34 sequence. These are 213 base pair inverted repeats which are interrupted by 8 base pair. Like you can see here that this is the green gene we are inserted between the two LoxP. When crossed with Cre, the gene get removed, and that's how it can create the knockout organism. So how the Cre-loxP work? By using the Cre-loxP system, we can do the inversion, deletion, or translocation. Okay. For inversion, when the Cre-loxP on two different direction, it may cause the inversion of the gene. Okay. If in the Cre-loxP sequence in the same direction, then it may delete the gene. It will delete the gene. And translocation, when there is two strands and there is possibility of exchange of strands. This is translocation, when the Cre-loxP is present on both the strands. So how we can use this Cre-loxP to create a to generate a knockout animal? First of all, we have to prepare the transgenic mouse which contain the LoxP, and another should have Cre. Like one mouse have the LoxP sequence and the gene in that, and another one should have the Cre, which should be under the control of promoter, which either should be tissue specific or should be inducible. Okay. When the mouse which have the LoxP sequence, LoxP and the gene, and another mouse which have the Cre under the promoter. Like in this case, there is example of nestin promoter. When nestin promoter nestin promoter Cre mouse is inserted with the LoxP, then it will remove the SCL gene, which is expressed in the brain tissue. And if we knock out this gene at the embryonic stage, then this is an embryonic lethal. The mouse will die. You will not get the animal to study. So that's the reason that we have to prepare some conditional knockout. If we will make a cross between these two mouse, then we will get a conditional SCLKO knockout mouse in brain, because this nestin is a pro-neural gene, and it is has specific expression in the brain only. So you will get a conditional. The another example of inducible system model, that here you can see that the Cre gene has been under the control of tissue specific promoter, and this Cre gene has been combined with the ERT. This is the estrogen receptor, and it is mutated one which can bind the TAM. What happens in the nucleus? This tissue specific promoter, this translate the gene, and this gene can export it out the cytoplasm, and after binding with the TAM, it get inserted, go back into the nucleus, and can remove the site, can remove the gene between the two LoxP site. So what happens if this is TAM is present, then only it will work. If it's not present, then it will not work. So it means it's kind of conditional system. If we want, we can do that. We can remove that, or if we see that it's like we can decide that time for that at what time we have to knock out this gene. So this is the benefit. This is the main advantage of using the conditional knockout system. There are many Cre-loxP based models. Like I have discussed, one is the Cre estrogen receptor, which work under the effect of TAM. This is a tamoxifen, and there are other inducers for Cre recombinase also, like Cre-ER is a progesterone receptor under the control of progesterone receptor. This is Cre-GR under the control of this growth receptor. In this way, one need to have induce these system using as written here. This is RU486. This is dexamethasone, and another system is MAX1 promoter Cre. This is inducible under the control of interferon. So this interferon based system, either to get it from the either use from the outside or from some tissue or through the infection. So this kind of system can work. Another is a TET based system, which is of tetracycline on and off, where when you presence of tetracycline, this system get activated. Let's summarize this module. We have talked about a knockout mouse, transgenic mouse, and the conditional knockout. The knockout mouse with a site-specific integration of transgene in the genome, where we have replaced the gene, endogenous gene, with the another gene, and the endogenous gene is completely knocked out. That is is helpful to create the knockout mouse. In transgenic mouse, we have done the random integration of transgene in the genome, and later we have screened that where in this process about 10 to 30% of the of the mouse of the generation mouse have like the transgene, and we can check them that where the what are the mouse which have the presence of transgene by the PCR. And conditional knockout, which is the tissue or time specific expression of the transgene, where the transgene is not expressed in the whole body. It is expressed either in some tissue or the time when we induce it. Thank you.